Welcome to Why Should I Invest? Developed by the International Collegiate Angels Network, or ICAN. At ICAN, we believe that campus-born ventures are today's most underappreciated asset class. Each podcast will spotlight a campus-born founder and the venture they're building. I'm going to ask the tough questions that investors would want to ask. And welcome to Why Should I Invest? I'm Dr. Tom Duening. Today I'm delighted to be with Dr. Brock Schuman. Brock is working on some very interesting things using a zebrafish model that is kind of fascinating. If you go to their website, azorbiotek.ca, biotech is spelled with a K at the end, you'll get a chance to see the video of Brock working with the zebrafishes. Brock, welcome to the show, and tell us a little bit about Azor Biotek.
Yeah, so I started Azor Biotek specifically to target microRNA. It's disease and target agnostic, but it was purpose built for targeting microRNA, which are the topic of last year's Nobel Prize in Medicine. And the reason we built it is that there are a lot of AI drug development companies out there that have totally transformed the drug development landscape, but they are incompatible with a lot of these hard to target drug targets. So we built a system that we think can. Yeah,
it's very interesting. And I was doing some research on the company and looking at the zebrafish model and so on. Tell us a little bit more about the AI approach, because that kind of is a buzz term. And
sure,
sometimes it even has a little bit of a negative connotation. But in your case, I guess what it enables is rapid prototyping of different drug models and different molecular systems.
Yeah, that's right. It's not a large language model. This isn't ChatGPT for coming up with drugs. This is using machine learning, using lots of data to try and train a system to come up with novel small molecules that are alien to anything anyone else is testing, which is important because if you want a novel drug, you need a novel molecule. And so there are a lot of these AI-guided drug development companies out there. They use different sorts of data, largely molecular data, and we are leaning into having the world's best phenotypic data. So seeing that actually affects a biological change in zebrafish and in bioprinted constructs, organoids. But it's really easy to see in the zebrafish if, say, your drug has cardiovascular side effects, because they're transparent and we can see their heartbeat and make an optical cardiogram out of that.
Right. And tell us a little bit about, because the video talks about this, how you're studying depression in zebrafish, which is something that, maybe would surprise people.
Yeah, well, that's really where Azor really got started out is I started off with this, it's called CanBind. It's a big network in looking at depression biomarkers. That's really what got me into microRNA. And so there were these microRNA that were important in treatment refractive depression, so people that don't respond well to antidepressants. And I thought, well, maybe we can drug them directly. So you can make antisense RNA that binds and blocks those overexpressed RNA, and it worked really good in the zebrafish models. It worked really good in rodent models, but I never thought it was going to be a good drug to bring to market, because it would have to be injected, and it would have to be injected right into the CNS. And psychiatric patients have enough going on; they don't need to go for brain injections every day. But zebrafish are interesting because you can't really model psychiatric conditions in them; they're too complex. But we could get some little, some hallmarks of depression in the zebrafish.
Right, and you guys use basically sexual frustration. You put a barrier between the males and the females, and they realized they couldn't get through the barrier.
Yeah, that was a fun model. So normally when you make zebrafish, and we're not using, just to be clear, we're not using this model in the present AI system, but I'll talk about it because it is fun.
Yeah.
Normally, if you make zebrafish, you kind of set them up in the same tank, the day before, males and females segregated behind a little barrier so they can smell each other and want to interact with one
Hmm.
another. And the next morning, after the mood's been set, you lift up the barrier, and eggs happen.
Yeah.
But what I did was just keep one individual separated,
Right.
and he could see what was going on. He could smell it. He wanted to join the party on the other side of the
Yep.
barrier, and we measured that they call it pharmacobehavioral
Yeah.
approach avoidance, which
Right.
declined over the course of a week.
Yeah. Interesting. And so you guys are doing a lot of research. I'm looking at all the different things. You say there's 10 different disease indications, in fact, that you've been kind of investigating.
Yeah, so we have 10 ongoing. The ones that are closest to clinical trial are one where it's not shared IP, but shared royalty with a paying
Hmm.
client. They've been a very good client. Um, for substance use disorder, and that's a big problem in North
Yep.
America for sure,
Yep.
or at least the English speaking
Right.
part of North America. Um, and our other one, and the ones that we're directly raising funds for right now, are anti-cancer therapeutics. So they're tumor inhibitors that target microRNA, and how microRNA works is it in turn silences a bunch of other transcripts, other drug targets, and ours silences 99 oncogenes simultaneously. So
Yeah.
we're seeing a really profound treatment effect when we're targeting it.
Yeah, it's amazing. I want to talk about your team in a moment, but let's talk about sort of the business model here, because I think there's a distinction between companies that are developing drugs directly and then trying to market the drugs. But it seems to me, look, from what I have, the information I had, that your business model is more around sponsored research. Is that a correct
Well,
characterization?
Well, and that'll change too. So we've
Hmm.
we've gotten some non-dilutive funding that has helped us get off the ground, and we've had some paying customers as well. We have some traction there. We've got
Uh,
about 1.5 million that have paid, and we've circled another potential 4 million in revenue
right,
this year. Once we meet certain milestones with the
right.
technology development. So that's where our revenues have come from so far, but that's going to be really asymptotic compared to the value of the drugs when they reach market. So what we
Hmm.
really want is to hold on to more of that novel chemical matter IP, and
Yeah.
that's what we're raising funds now for. So we have all the IP around the tumor inhibitors, and we're raising funds to enable bringing that to clinic.
Right. And then that would be done basically through a licensing type model.
Well, we've been reached out, so you can license it. Yeah, eventually we'll have to
Hmm.
license it to big pharma, or we can get a faster returns if we do preclinical licensing. And we've actually been approached, kind of. We weren't looking for preclinical licensors, but we've been approached from both American and European markets, people interested in licensing our technology.
Yeah. And you've got a fascinating team on board. Why don't you
All
tell
right.
us a little bit about, a little bit more background on yourself and a little bit about the rest of your team?
Right, yeah, and we're going a lot of active growth right now. We've been 2 years ago was just me, and now we're up to 10.
Oh,
So
right.
we're
Yeah.
growing fast.
A real company. Yeah.
But the three founders with the most background, or the three founders are me, and I've spent over 20 years writing software for mostly for structural biology, but scientific software. I've kind of gotten into machine learning kind of sideways later in the game. Our other co-founder, Javier Alfaro, who has a professorship at the University of Calgary, has a more formal training in biomedical AI. He came out of the same University of Toronto lab as Atomwise founders. Atomwise are they're valued at I think $740 million last time I checked. So his training is good. So, in addition to writing software, I have a PhD in biochemistry, and I have spent about 15 years now genetically modifying zebrafish for drug development. So it's
The
that's kind of our
zebrafish is your friend, right? I mean, you know the zebrafish pretty well.
yeah. I'm a
Yeah,
big advocate, and
yeah,
one of the reasons to those who aren't initiated, above and beyond being transparent, so you
yeah,
can see internal systems working really well. They're very cheap. They're 1/1000th the cost of a mouse. They're even cheaper than cell lines, you know.
yeah,
And they're a whole organism. So if you're
yeah,
looking at a cell line, you know, it doesn't have a liver, it doesn't have a blood-brain barrier, it doesn't have skin. So you're looking
right.
at a whole systemic response to the drugs rather than
Yeah,
hoping that this one cancer line will be representative of a whole organism.
right.
And
They
I
lay a
should I
lot of
should
eggs.
mention our
They lay a
oh
lot of
yeah,
eggs, and
yeah. A breeding pair makes 200 eggs, so that's a lot of material.
yeah,
Perfect. I neglected to mention our COO, who I should bring up,
right. Please do.
Lubos Merrick, because you know I started off without any business background, and I've just been kind of learning it going along. But Lubos is very experienced. He has had leadership positions at Takeda, Novartis, and Alexion. Alexion is noteworthy because it was AstraZeneca's largest purchase to date, and he helped negotiate that $39 billion sale.
Yeah, cool. And then Javier, and you've got some investors on board as well.
Yeah, that's right. Yeah, we got our first two investors earlier this year.
Mm-hmm.
Those both came out of a life science accelerator we were doing
Mm-hmm.
out of Alberta. It was called TNT Life Science. So that was
Right.
a great program that I'd
It
recommend
is.
to anybody in Western Canada. Yeah,
Yes, it is. Yeah. And you raised 100,000 from your investors.
yeah, that's right.
Uh-huh. And your non-dilutive funding, is that coming from federal organizations that NSF or something like that?
Yeah, there's a Canadian equivalent, which is IRAP. That's where
Okay.
most of the funding has come so
Okay.
far. And then, yeah, that's been our major non-dilutive source so far.
Okay. And you're currently in a round, and you're seeking 100,000 to 4 million, according to my information that I had in front of me. That's a big range.
Yeah, yeah, that's right. Well, I mean, we're seeing a $4 million raise, but
Right.
we'd accept 100,000.
Yeah. Right. And what do you think is the use of funds on this raise?
So about half of those funds would go to what's called the investigative new drug
Yeah.
development, kind of getting those tumor suppressors to clinical
Uh-huh.
trial, and then the other half would be kind of for internal growth as well as starting some other projects and
Right.
technology development, but. About half go to this the tumor suppressor development.
Yeah. And, one of the things that investors always freak out about is when you talk about the FDA and the phase 1 trials and everything, and how
Yeah.
long, and they just think how long that's going to take. But I have a note here that says that the AI discovered molecules achieve 80 to 90% success rate in phase 1 trials, which kind of speeds you through the process.
Yeah, well, I think a big part of it is it cuts down on the cost. When it comes to clinical
Yeah.
trial, we do have a pretty good strategy in that even though our tumor suppressors are indicated for a number of fairly common cancers, but if you try to do a clinical trial on a common cancer, it's going to be a
Yeah.
very expensive and long-term project.
Right.
So we're focusing on glioblastoma, which it's
Uh-huh.
also indicated for, and which we have a very good zebrafish model for. And glioblastoma is a brain cancer. It's a fairly common childhood
Right.
cancer. And doing that, we anticipate being fast-tracked, and it's a novel mechanism.
Yeah.
So, we'll try to be as aggressive as we can to
Okay.
get it through quickly, but it should be relatively cheap and fast. But even before reaching clinical trial, we'll have a value reflection point for investors
Yeah.
in case they start to get cold feet.
Yeah.
But I think we'll only see good results, and people will be more interested as things
Yeah.
go on. But there will be value reflection points, even without any kind of licensing. We anticipate about fourfold growth within the next 2 years.
Right. So let's talk about your differentiation. There's a lot of drug discovery firms out there, as you mentioned, some of them also using AI for the rapid prototyping and, and modeling of different molecular systems and so on. But you say you're the only ones that have RNA targeting, clinically relevant training data for your RNA targeting. Tell us more about
Yeah.
that. Yeah.
So there's two caveats there. So there are other AI companies that are trying to target RNA.
Yeah.
Our philosophy is that their approach isn't going to work.
Mm-hmm.
And there is another really well-established company. Uh, oh, Jeepers.
Yeah.
No, sorry.
You're blocking on it. That's okay.
No, that's all good. I have to look it up now. I have to remember.
Mm-hmm.
Recursion, right.
Okay.
So
Yeah.
Recursion are really well established.
Uh-huh.
They're valuated at nearly $2 billion, and they use a lot
Okay.
of really good molecular data. And they have these phenotypic profiles that they're using to train their AI,
Right.
which is good. But their phenotypic profiles come from those, 2D cell cultures. And so, it has limited clinical viability,
Mm-hmm.
whereas we're looking at real clinically viable things. Like we're looking at, pharmacokinetics. We're seeing if the drugs make it to the
Right.
brain. We're looking at cardiovascular side effects. We're tracking individual tumors and whether the progression of those tumors, if the drugs we're adding makes them smaller, that
Yeah.
are in the brain, so hard to reach.
Yeah.
So we have, I think we're setting a new standard for
Yeah.
clinically relevant
Okay.
data to train that
Right.
system.
So I love this story. It's a great story. I think you're clearly in a great spot. Let's talk a little bit about the market then. So our customers at this point are other drug distribution companies, drug development companies that are hiring you on a contract basis to do your research, and then do they own the intellectual property that you create for them?
Well, for those customers, it can go one of two ways, and we're
Mm-hmm.
happy either way. So we have one customer who are not traditionally in drug development.
Mm-hmm.
They have a cash positive model for opioid addiction recovery,
Yep.
where they fly people with addictions down to destination tourism clinics to get
Mm-hmm.
therapy that's
Sure.
has not traditionally been legal in the United States. And so we're developing molecules that'll be safer. And because they don't know anything about drug development, we're keeping all the IP, but they will get a share of royalties when it gets to market.
Right.
But we're happy to go the other way. We're happy to give IP to the customers if they want to develop it, as long as we get a share of the royalties if it makes it to market,
Yeah.
and milestone payments as
Right.
well.
Right. And how many? And then you also say you have a B to A business model, B to A, which I guess is
Right.
business to academe.
Yeah, that's right.
Yeah.
That's right.
Yeah.
Yeah, yeah.
And what
So
does that
that's
look like?
so we have I didn't talk about it in too much detail, but we have this high throughput kind of innovative imaging system. Nobody else is training their AIs with this kind of imaging system. It
Right.
really is kind of groundbreaking, especially. I won't get into too many details,
Yeah.
but we wanted to house it at the university
I
so
see.
that as many people who could benefit from it can have access to
Yeah.
it. So it's kind of parked at the University of Victoria.
Right. And so you're going to
And
be bringing in more scientists,
yeah, so
more AI experts, and
so
other
we
kinds of things.
anticipate,
Then what would be the exit
at least
for a company like this?
paying for its upkeep through user fee to people at the university.
Yeah. Sure.
Yeah.
Right. Yeah. Right. Right.
Yeah, I think that's absolutely
Right.
right. I
Okay.
think
That's fine. And then, of course,
I
I
think
always
that's
end
great. And any
this
AI is
podcast with
only
the
going to
simple
be
question: why
slightly
should I
worse than
invest?
the data you train it with. And we are just trying to use the best data so that we can get the best drugs out. Yeah. Well, my vision, which is that within 5 years, we
Wow.
hope to be
What a great
start getting
story,
royalty
Brock.
payments, and we can
Congratulations on
stop asking
building
people
this
for
this
money
really
and
interesting
just
company.
develop
I'm
drugs
fascinated
for
by
as
what you guys are
many
doing. We certainly
disease
need
indications with unmet
more
needs as possible. Because
of
the
using
technology
AI
is
in the drug
disease and
discovery
target
process. And so
agnostic.
you guys are
So
leading the
we
way,
hope to
and I
do a
appreciate
lot
you
of good
joining my
things
show
with it.
today.
Um, I think I struggle with the word exit sometimes. But I think definitely when there's for investors, there will be an opportunity for an IPO, or there will be an up like down the road, not right away, but there'll be an opportunity for an IPO, or we might get we could sell to some large pharmaceutical industry, but who knows? Maybe we'll grow larger than some of the large pharmaceutical industries. Will be more of a permanent fixture. I think people should invest in me because we're building the new standard in biomedical AI, and this is your opportunity to help pave the way. And if you have disease indications of interest, we can certainly start those research streams. Okay, well, thanks so much for having me, Tom.
For more information about how you can get involved in the ICAN mission, please contact me directly via email at tomdenning@icanhub.net.